Hi there. So I'm going to talk through with you today how to get started on your AB assignment. So this is your assignment brief. So if you open this up, try and save a copy into OneDrive. It's just a little bit more secure in OneDrive, automatically saves, easy to access from home, etc. First of all, you must fill in the orange boxes on here. So fill in your name just here, your teacher's name just here, your date the assignment is submitted. Now you've got 2 weeks to complete this until the Thursday at the end of term. So the submission date is going to be the 28th of March. Okay, in these boxes here, you're going to write down which pages you have written about each marking criteria. So that's something you probably want to do at the end of each section or at the end of the whole lot. You need to write your signature here. You can just write your name on the computer. So I would write H.A. Val, for example. That's fine. You don't have to scan in a signature or anything like that, and then put the date that you filled that in. Then down here, you need to put your assessor name again, your handout date, which is today, 15th of March, and your hand-in date, which again is 2 weeks, Thursday, so will be the 28th of March. Okay, so your vocational scenario or context. You've obtained a work experience placement within the microbiology lab of a large teaching hospital. The department is in the process of producing educational materials for patients, student nurses, and other trainee hospital staff to illustrate the threats to human health resulting from infectious diseases caused by bacteria, viruses, fungi, protozoa, and prions. As part of this initiative, you have been asked to produce an illustrated portfolio that details the methods used to classify pathogenic organisms. Your portfolio must also include factors that affect the growth and virulence of microorganisms, the pathogenic mechanisms that lead to the development of the disease in infected individuals, and the strategies used to treat different infectious diseases. So we're still on that work experience placement, and we're now producing this write-up. So then the next section goes through the tasks. So you can look at these tasks and follow them through if you wish to. But what we have done for you is we've summarized all of these pages here into one document, which looks like this. So what I'm going to do now is I'm going to talk you through this document, which tells you what you need to write up for this assignment. There's going to be a few of these videos for each section. You're going to write up your work just underneath this table here, as you did before. So literally you can just go onto new lines underneath here and start your write-up underneath this table. So what are you going to include in your assignment AB? So as the name suggests, it consists of a part A and a part B. Now this video is all about part A, so we're starting at the start. Now this document here, which you might have seen, has part A split into two sections. The first section is classifying microorganisms, and the second section is the virulence of microorganisms. Now what you need to write for pass 1 and merit 1, or pass 2 and merit 1, is basically the same. Pass just means less detail, merit is more detail, so we're all aiming for merit, so there aren't separate sections for these. So the first section here is about how do we classify bacteria. So we've got a bullet point list of things that you're going to include in this section. And for each bullet point, you need to explain how these differences make the bacteria clearly different to each other. Okay, so what we're going to do is we're going to write a whole paragraph on each bullet point. Okay, so within bacteria, we're writing a whole paragraph, a whole section on each of these bullet points. So the first bullet point that you need to write about is the organelles that you find in bacteria. So if you go into your assignments and you go into assignment number 1, assignment AB, part 1, where's it gone? Let's find it on my list. Set it a long time ago, didn't I? Here it is. Okay, so if you go into this assignment, what you'll find is that, oh, I think you should have some attachments on there. Let's try another class. Maybe that class didn't get the attachments for some reason. Let's double check. Try this one here. So you should have the PowerPoints attached. If you haven't, just let your teacher know, we can change it. Or maybe it was just lower down and I missed it. So attached to this bacteria fact file homework, you will have had these PowerPoints. So first of all, if we look at classification of prokaryotes, and you open up that PowerPoint just there, you'll find a lot of the information you need for your write-up in here. Now, you might find that you've already written some of this up in your fact file, in which case, copy and paste the sections across. That's absolutely fine. So the first section is about the organelles that you find in a prokaryote. Okay, so if we look at a diagram like this, this shows us the organelles. So you need to be writing a few sentences about each part. So what kind of what is the chromosome? What's the, is it linear? Is it circular? Is it made of DNA? Is it made of RNA? Is it in a nucleus? Things like that. So find some detail about the chromosomes found in prokaryotes. Find some detail about what the pili are for. Write a bit of detail about ribosomes are for, a bit of detail about food granules, a bit of detail about flagella, plasmids, cytosol, plasma membrane, cell wall, and capsule or slime layer. So they are the organelles. They're what you're meant to be talking about in this section just here. Okay, so if you look through these slides, you can find some slides to help you, but equally, you can find information on the internet as well. Okay, so I'm just trying to find a good section in here. So look, if you go up to page 26, up to 26 was a bit of an overview of all the organisms. So from page 26, we've got specifically prokaryotes. There's some resource links here as well that you might want to click on and get information from. You've got a video there, which is good. I don't think you've got anything more about organelles here, so you might want to just use that diagram that I showed you and look up all of those parts to enable you to write your paragraph all about the organelles. The next section you're going to write about is the shapes. So in back to your PowerPoints or back to your fact file homework that you did, there's definitely some bits about shapes of bacteria. So the main categories of the shapes of bacteria, it's also called morphology, is spheres, rods, and spirals. So you probably want to include a diagram, reference where you get your diagram from, and talk about the different shapes. You can talk about how there's more than one example within each as well. So even though the spheres or cocci is a category, you've got different types within that. And even though the rods is a category, you've got types within that. And spirals is a category, types within that. So you've got these subdivisions, which you can talk about as well. And that makes bacteria different to one another. The next one is appearance in large numbers. So lots of bacteria are not found on their own. So you can talk about how staphylococci are always found in these bunches, almost like grapes. You could talk about streptococci and how they are always in lines. You could talk about chains of bacilli. Okay, so sometimes we can tell bacteria apart because some of them are on their own, some are in pairs, in fours, or in larger groups. The next one is the reaction to the gram stain. You're quite familiar with this because you did your gram stain reaction in assignment C, but you've got some information on here about how gram staining works. So you need to link it to the cell wall structure. So you need to know that the gram-negative bacteria have a different strong cell wall to the gram-positive. You need to talk about what the difference is. A diagram would be great. Reference your diagram. And then you can explain how the gram stain is done and why the gram-positive end up purple and the gram-negative end up red or pink. Okay, why they end up different colours. And you can add that in. Use the PowerPoints and then look at more detail yourselves. The next section is we can classify bacteria based on their oxygen requirements. So oxygen requirements in general, you could talk about aerobic and anaerobic, but then we have got these subdivisions even within aerobic and anaerobic. So you've got some good keywords here that you can use to do a little bit more research and write a paragraph about the oxygen demands and how different types of bacteria have different oxygen demands. The next part is the temperature requirements. Now, the temperature requirements isn't in that PowerPoint. The temperature requirements was in lesson 2. So some bacteria are adapted to live at high temperatures, some are adapted to live at low temperatures, and there's definitely a slide in here where we went through thermophiles, mesophiles, and psychrophiles. So you'll have written those down in your booklets. Obviously, you can look them up on the internet as well. And there are 3 different types of bacteria that are adapted to live in different temperatures. Now, you could include in this one a little temperature graph, and you could explain how different bacteria have different optimum temperatures, which is where our graph has its highest point. And you can talk about why they can't survive when they're too far from their optimum, because if they're below the optimum, the molecules have less kinetic energy and we don't have enough successful collisions for reactions to happen, stay alive. And if the temperature is too far above the optimum, the enzymes denature, and you could include what's meant by that. Then you're going to go on to your next section. So your next section is classifying protists. So for protists, you're going to need to go into a different homework to get your resources. So you need to go into your assignments and you need to find the homework fact file that you did. That was on protists. So I need to save that. Go back, find your homework 2, which was on protists, and yours should look like this. So you've got the PowerPoints that link to these lessons. So if we go into lesson 6, we've got some information about protists and fungi, which are both types of eukaryotes. Okay, so the first way that we can distinguish between different types of protist is by the way that they move. So protists can move in all different ways. Let me just find which bit you're going to be using. Here we go. So movement of protists means their motility. And we've got these different methods of locomotion that you need to talk about. So we've got some that move with these false feet or pseudopodia, like amoeba. We've got some that have got like a tail, like a flagella, and some that move with cilia. So they're your main categories. There's a few more keywords you can look up here. There's a good video that you can use there. And you can talk about how different protists move in different ways, and that can be used to classify them. The second way we can classify protists is based on their organelles. So the organelles that protists have, if we go, I think, back a little bit, maybe. So here's a diagram, for example, of a protist. So you could use this diagram of the protist, find your own diagram of protist, and you can see some of the organelles. So all of these labels here are organelles, and you don't want to just list them. You want to write a sentence or 2 about each one, describing what they're for, how are they adapted. So we can classify different protists because they have different organelles. You can also talk about what they have different types of nucleic acid and what they might be. The third way we can classify protists, your next paragraph, is by how they reproduce. So protists can reproduce sexually, asexually, or both. Okay. So you need to find out a little bit about this. That goes into fungi. I'm just seeing if there's anything about protists reproduction. Anything more about that? Fungi. We're not on fungi yet, that's why I'm skipping those. I don't think there's much info on the slides apart from this one, but there is a video just there. Okay, so for that one, you're going to have to go on that video link and get your ideas from the video and then do your further research from the video there. Then you're going to move on to fungi. So fungi and protists were in the same PowerPoints because they're both eukaryotes. So first of all, how do fungi reproduce? Now, fungi kind of fit into 2 categories. Some types of fungi are the ones that have mycelium, and some types of fungi do not have mycelium. So we have got different types of fungi, and we can use their method of reproduction in order to characterize them. So there's a little bit of information about what mycelium fungi are and about what non-mycelium fungi are and how they reproduce. So you can see here, the mycelium fungi make spores that are released and spread, okay, to grow new mycelium, whereas non-mycelium fungi multiply by budding. So you need to have diagrams and explanations of how these mycelium type fungi produce their spores, and diagrams and explanations of how things like yeast multiply by budding. So they can multiply in different ways. Okay, there's a few diagrams here about fungi which might be helpful. The spores can either be sexual or asexual, and the budding is always asexual. You've got lots of slides to help you with the fungi reproduction section. It's nice to have these diagrams and use the information on those. And I think that's about it for that one. Fungi can be classified based on their internal structures. So the structures of fungi, this is quite a good diagram. We've got some simple diagrams here. You can see the structures in the fungal cells there, the reproductive structures just there, and you can see some more detailed structures of what you might find in fungal cells just there. So you need to talk about the structures inside the fungi and how they could be used to classify different types of fungi. And then the next section is the temperature requirements of fungi. So different fungi are adapted to live at different temperatures, and you might be able to find some examples of that as well. I'm just wondering if there's anything more in this one relevant, because a lot of it's going to be used for the next sections. You see, you don't have to use all the PowerPoints in this section. Right, nothing there that you need, that's fine. Then you're going to go on to your viruses. So then you're talking about viruses. Remember, paragraphs for each section. So you need to find your virus fact file homework. So go and find your fact file 3. Again, if you've got bits already written up, that's great. Just use what you've got, copy it across, add to it. So for, oh, that's not right. Oh, there's more. The fungi was separate fact file. We want fact file 4 for the virus. Apologies. So here we go. So here's our PowerPoints that we can use to help us with the viruses. So the first section for classifying viruses is the shape. Now viruses come in lots of different shapes. So we've got a few examples here. We've got a few different shapes shown just there. There's a good slide here about the shapes of viruses. So we've got helical, polyhedral, spherical, complex. So diagrams would be really nice. Photographs from microscopes are nice. Reference where you get your photos from and talk about what are the characteristics of each of these shapes. An example of each would be great. And compare how you could classify them based on those shapes. Some viruses have an envelope. Okay, so if they have an envelope, then they can be either without an envelope, they're called naked, or with an envelope, they're just called envelopes. So explain that difference again. There's no separate, oh yeah, there is, sorry. The third one is genetic contents, which is on this slide as well. So the genetic material can be DNA or RNA, and you can talk about that. You might want to add in a bit about retroviruses with the RNA and how they need to have reverse transcriptase to make DNA so that then they can get into the host cells. You can talk about the genetic material being single-stranded or double-stranded, linear or circular, segmented or non-segmented. Try and find out what all of those terms mean and explain them. Don't just list what's on the PowerPoint. You can talk about the size of viruses, how different viruses are different sizes, and how that might help to classify them as well. Now, for classifying viruses, you might want to add in some detail about Baltimore classification, and this goes with your DNA section. So depending on the type of mRNA that they produce when they multiply, they can be classified in a Baltimore classification system. So link that into your nucleic acids section for this one here. Viruses can be classified by their type of host as well. Let me see if I can find anything about hosts of viruses. I've just noticed there's nothing about the type of reproduction on here. There probably should. I think I'll add that on. Viruses do replicate in different ways. So they can replicate via the lytic pathway or the lysogenic pathway. So it'll be really good to have a diagram of that and explain those two methods of reproduction. I'll add reproduction onto your checklist. Viral reproduction is an important way of classifying the diseases as well. Now, when it comes to the host that they infect, viruses can infect all type of organisms, so animals, plants, fungi, bacteria, and prototists. So when you talk about classifying viruses, you can say that they're classified based on what it is that they infect. Try and find some examples of what viruses infect different things and how the difference is there. Then we're on to the last section of this section 1, which is classifying prions. So first of all, prions can be classified by their host species. So the second part of this PowerPoint went on to onto the information about prions. So we've got an example just there, a little bit about classification here. So classification can be done based on the host, what do they infect, people, cows, etc. They can be classified by the disease they cause or by the type of damage or lesions they cause. So there you're basically lining up with those headings just there. Really usefully, there is some an internet link here to a good website that you can use to help you on that. I think that's about all from that one. See if there's anything else useful on this one. So virulence is the next section. So we're not using the virulence very much in this section. Just looking for prions. No, I don't think there's anything you need in that one there for prions. There's much information in the PowerPoint really for prions, so it's going to be up to you to do a bit more research. So just be really specific in your searches. So what are the host species that prions affect? Different types of clinical diseases. I tell you what there is on here, however, is the examples of prion diseases in the number 8, lesson 8 at the bottom. There was on this one, there's a prion disease here for CJD, so you can look up that. And scrapie, you can look up for prions as well if you need a starting point. Okay, I'm going to finish the video here, and I'm going to do a separate video for section 2 and then another separate video for part B. Okay, so you need to follow a few videos through. All right.